Okay, hi everyone. My name is NUREEN AFISHA binti ADNAN. My matrix number is 208291.
ANATI, is your turn now.
Sorry. Okay. Hello. My name is ANATI NABILAH binti TAJUDIN SHAH, and my matric number is 205706.
Team go first or me?
You first.
Oh, hi, my name is ZARIFAH binti AZAMUDDIN. My matric number is 207809.
Hello everyone, my name is Timothy Lee Zhenzhou and my matric number is 207980.
Hello everyone, my name is GUAN. Matrix number is 206974. So today we're gonna discuss the practical 3, which is our surface sterilization of leaf expand result and the discussion. So without further ado, let's look at the first question. Okay, the first question is all about, based on the observation, explain the result obtained, suggest a modification, if any, to improve the protocol. So Rifa, can you answer that?
I want to share screen. Okay. Can you see the picture?
Okay.
First, our task, we need to use the mercury agent, sterilized agent, so 0.5%. The exposure time here is 1 and 5 minutes. And number of explant, I put 5 on 1 minute explant. And here the result. So the number of contaminant, I put 3, and the number of viable explant, I put 2. So the percentage that I got is 60 and 40%. The way I got the percentage is 3 over 5 times 100%. And the number of the explant is same with number of contaminated. So it's 60%, and the number of contaminated is higher than the number of viable explant. So it's a contaminated plate. For a 5 minute exposure time, I use 3 explant, and 1 for viable explant and 2 for contaminated explant. So I got 33.33% of contaminated plant and 66.66 viable explant. So the number of the explant is 33.33%. So the number of contaminated is higher than viable explant. So it is contaminated. It's not contaminated plate, as the number of that plant is lower than viable explant. Sorry.
Okay, so Rifa, based on your result, do you have any modification?
I think no, but just a reminder, we use the mercury agent. So the important thing is always to wear a glove and always to wash your hand after you using the glove. And don't put the glove anywhere, because the mercury agent is so dangerous and it could harm your skin and environment. That's all, I guess. So for the second question, which is the conclusion, I drew this, which is say that the number of contaminated plant is higher if you use only 1 minute exposure time. Are you agree with my conclusion, or do you want to add something?
Yes. Before look into the conclusion, I would like to discuss the first question. Yeah. So we know that at the step 6, right? We're gonna put all the things into the laminar flow. So I think to improve the protocol, I think that to ensure everything that need to place inside the laminar airflow should be sterilized first. For example, wipe the surface of the beaker and then some of the apparatus before moving into laminar airflow. Yeah. So anyone got any suggestion for the first question modification? All right. If then we look into the second one. So for my answer is actually so the higher the sorry. So my conclusion is the longer the time, okay, the more the effective the sterilizing agent, but at the same time it will reduce the viability of the plant. Yeah.
Thank you.
Okay. So everyone want to add on? If then everyone okay, then we move to the third question. So ANATI.
First of all, I don't have anything to share because I just wrote it down on my book. So I'm just gonna say it. For question 3, the question says, apart from exposure time, explain other factors that may affect the efficacy of the sterilizing agent used in this practical. First factor that I found is that the physical chemical environment, such as pH or pressure. Increase in pH will improve the antimicrobial activity of the disinfectants, while reducing the antimicrobial activity of others. The pH affects efficacy by altering the disinfectant molecule of the cell surface. The second one is temperature. Some disinfectants can increase its activity when exposed to high temperature, but there is also disinfectants that when you increase the temperature, it can degrade and weakens the germicidal activity and thus might produce a potential health hazard to us. The next one is presence of organic matter. Organic matter can interfere with the antimicrobial activity of disinfectants. Interference occurs by chemical reaction between the germicide and the organic matter can result in a complex that is less germicidal or probably non-germicidal at all, leaving only less of the active germicide available to attack the microorganism. And the last one is the type of microorganism that can be found on the surface of the explant. Community of microorganisms that adhere to the surface and surrounded by a complex matrix of extrapolymeric substance is known as biofilm. The biofilm, such as bacteria, and fungi, can induce microbial resistance to disinfection. In this case, chlorine can be used to break down and helps to remove the biofilm. That's all for question 3. Anyone wants to add any more?
Yeah, I want to add on. Yeah, such an interesting many facts from the sharing from ANATI, and I got one point that you guys might not think of is the type of the plant leaf. Imagine not every plant leaf has same structure, right? The physical structure, some thicker or thin. So maybe some of them have a thicker wax, cuticle wax. So it may affect the diffusion rate for the sterilizing agent or the surfactant and some such more things like that. So yeah, so that's my point. Anyone want to add on? Okay, if then everyone is okay, then we move into number 4.
Question 4. If the surfactant is not used, predict the outcome of the particle based on the percentage obtained above. For this question, I'm not quite sure, but I think the percentage would either remain the same or decreases. This is because the purpose of using surfactants is to reduce surface tension and allow the chemical agent to react with the whole surface of the plant. Since surfactant is not used in this case, I believe that some part of the plant would not be, some part of the surface of the S plant would not be penetrated by the chemical agent. That's why the percentage decreases. Yeah.
Yeah, everyone has same answer, right? The percentage of contaminated plant will increase, right? Yeah.
Yes.
So it's an easy one. So number 5.
If the step that treated the S plant with 17% of alcohol was ignored, could the outcome of the particle be changed significantly as plain? Okay, if the 70% of alcohol was ignored, I think the outcome can be changed significantly because the number of contaminated and that dead S plant may increases. This is because the alcohol is used to help kill microbes, bacteria, and other microorganisms on the surface of the S plant before transferring into an in vitro environment. I believe since the alcohol is ignored, the probability of the S plant being contaminated while transferred into the in vitro condition is high. That's why I think the outcome can be changed. Yeah, that's why. Anyone is
Yeah, and I think you're right. Besides you talk about that, I think that the difference between the alcohol and the Clorox one is a protic and another one is a aprotic, right? So in this case, the alcohol actually much more effective when it contact with organic matter compared to the Clorox, and it's actually the Clorox will deactivate by the organic matter. Yeah, so it will reduce the efficiency of the sterilizing effect if we didn't remove the organic matter first with the alcohol. Yeah. So everyone want to add on? If no, then we move into question number 6. Nuri, it's your time.
Okay, thank you, Guan. So question number 6, suggest a suitable sterilizing agent to replace Clorox used in this practical and modify the protocol to suit the new sterilizing agent. So I think another suitable sterilizing agent to replace Clorox use is 12% hydrogen peroxide solution. So to modify the protocol to suit this hydrogen peroxide solution, we should not expose the hydrogen peroxide solution to the air for a long time as it is relatively unstable and may decompose to water and oxygen. So, and then the exposure time of the leaves to the sterilizing agent is only 5 to 15 minutes. My another point is that maybe we can replace Clorox using calcium hypochlorite instead of sodium hypochlorite. So maybe we can increase the concentration of calcium hypochlorite. It's just like Clorox, it has two ingredients, but which are calcium hypochlorite and sodium hypochlorite. But usually the concentration of sodium hypochlorite is more than the calcium hypochlorite. So maybe we can increase the concentration of calcium hypochlorite instead of sodium hypochlorite. Okay. For this question, that's all for my answer. Anyone has another answer?
I also use hydrogen peroxide. Yeah. So the protocol is just a little bit change. You know, the hydrogen peroxide is very reactive, and then we usually store the hydrogen peroxide in a very cold place, right? So when we move out the hydrogen peroxide, we must use as fast as we can before it oxidize. Yeah.
Yes, exactly. So move on to question number 7. Name the type of sterilizing agent used in this practical. I answer halogens because sodium hypochlorite is a halogen. Any other answer? Same.
Yeah, for just now, Rifa say is the mercury chloride, right? I think that the heavy metal also play a role in the sterilizing effect. Yeah. So I very confused in this question now. What I answer is chemical method. I don't know. You guys got any different answer beside halogen or chemical method? It's okay, right? Okay, then
No.
to the question number 8. Continue.
Okay. Thank you. So next, question number 8, name the active ingredient of Clorox and its concentration at 10% volume per volume solution of Clorox. As I mentioned before, the active ingredient of Clorox is sodium hypochlorite at the concentration of 0.53% at 10% volume per volume solution of Clorox. Everyone agree with my answer? Yes. Okay, so
Yes.
thank you. That's it.
So thank you, Nureen. So I'll be answering question 9 to 11. So question 9 is explain the procedure to prepare 10% of Clorox solution and mention whether do we need to autoclave the solution or not. So before we start preparing our 10% Clorox solution, we have to remember some precautions. So for accurate measurement of the amount of bleach added, we can use a measuring cup. And as we know, sodium hypochlorite decomposes with time. To ensure its effectiveness, it is advised to purchase recently produced bleach and avoid overstacking. And clean water or ethanol should be used to clean away the bleach solution after disinfecting the equipment. So before we start preparing our diluted bleach, we must know that bleach is a strong and effective disinfectant. Its active ingredient, just like Nureen has said, which is the sodium hypochlorite, denatures protein in microorganism and is therefore effective in killing bacteria, fungus, and viruses. And all the equipment used must be soaked in diluted bleach for 10 minutes. So we have to check the first step, we have to check the concentration of our bleach bottle, and second, we have to keep all the windows and keep the windows open when diluting or using bleach to ensure good ventilation. And we have to remember to put on protective gear when diluting or using bleach, as it irritates mucous membranes, the skin, and the airway. And cold water should be used for dilution, as hot water decomposes the active ingredient of bleach and renders it ineffective. chlorine solution gradually lose strength, and freshly diluted solution using 5.3% must therefore be prepared daily. So we know that to prepare to to prepare a diluted bleach solution, we know that it's a 1:10 ratio, which is 1 liter of bleach and 9 liter of clean water. So and the next step, we use the plastic containers for mixing and storing bleach solution, as metal containers are corroded rapidly and also affect the bleach. And we know that bleach decomposes in heat, so we have to store out of direct sunlight. And wait.
It's okay. Then I ask you, should we autoclave the Clorox not?
We should not autoclave the Clorox, because when we autoclave Clorox, the Clorox will produce a toxic product that will quickly corrode the autoclave. And we should also never mix with other substances like because never mix. We should never mix the Clorox with chlorine solution or store them with cleaning products, because it contains ammonia, ammonium chloride, and ammonium chloride and phosphoric acid. So, does anyone have any answer?
Okay.
No.
Very good. Move on to the next.
So question 10 is to justify the need to rinse and expose the explant with sterile water for a certain period of time after Clorox. So when the explant has been surface sterilized, it is usually removed from the sterilizing solution and rinsed several times in sterile distilled water. Because during the sterilizing, because after exposing the explants to sodium hypochlorite, we must rinse them to remove all traces of mineral from the explants. And I think that's all.
So can I ask you why should we rinse or wash all away the sterilizing agent before we move into further step like incubate them furthermore?
So that is to I didn't find the answer yet.
Never, I have your answer. Because actually we know that the Clorox are actually very strong sterilizing agent. Yeah. So if we don't wash away, right? It actually will kill the plant. You know, it will penetrate the cellular component and then kill that. So that's why we must wash away before we move into further step. Yeah.
So question 11. That's it. So question 11, if rinsing with sterile water was performed quickly and without leaving the explant in water for a while, what will be the consequences, and predict based on the percentage obtained above. So in my opinion, because it is important to like carefully sterilize the plant so that there will be no cultures that become contaminated with bacterial or fungal growth. And since the question here says it was performed quickly, so in my opinion, I think there will be some contamination and will ruin the result. Will ruin the observation. And the percentage will the number of contaminated explant will be increased, and the number of viable explant percentage will decrease. And does anyone have any answer?
Oh, I actually have different answer. Because actually the rising with sterile water, just like the question 10, right? So actually my answer is the number of dead plant will increase and turn brown. Yeah, so because got the leftover Clorox. Yeah. So everyone want to add ons? All right. So thank you, Timothy. Okay, now it's my turn. Now it's my turn. So the question say that, okay, if the source of the explant was not heavily contaminated, which part of the sterilizing protocol could be modified to make it more effective? Explain. So if it is not heavily contaminated, I think that we can skip step number 6, which is the immerse into the 70% of alcohol. So I think that if it is not heavily contaminated, maybe that the leaves are not covered with the dirt or some any matter. So I think that skip for this alcohol step can accepted because the next step will be immersed with the 10% of Clorox, which has same function as the alcohol to sterilizing. Yeah, so that's my answer. Okay. So if everyone agree, then I look into the question number 13. So he said that if the source of explant was sterile, which part of the protocol could be modified? Explain. So I think that this question is, I don't know if funny or what, because if the explant was sterile, which means that it can directly cut and then to further incubate, right? So I think that we can skip the alcohol part and the Clorox part. So if we skip these two, then I don't know that the data recording can we do or not. So yeah, so that's my answer. If the explant was sterile, just can skip the sterilizing agent and just directly cut and incubate there. Yeah, so that's my answer. Everyone want to add on? Okay. So in question number 14, it says suggest a method to grow a plant under sterile condition for explant preparation. So what I found on Google is actually one way to grow the explant in the sterile condition, which are the micro propagation. Yeah, so the micro propagation is actually what it's a practice of rapidly multiply stock plant material to produce many protege plants. Yeah, it sounds very general. So it's actually, I will explain step by step. So it actually got 4 steps, which are which are one is the, taken out preparation of the explant, and then the second one is the growing of the multiplication of the explant, which multiply through the shoot by using the stimulating hormone. And then the stage 3 was the rooting and the azamatizing, which are the using the also the stimulating hormone. And then the step 4 is the transfer new plant to soil. So my answer for these are very limited. Yeah. So do you guys got any other answer? No? Okay, if then I think that we have reached our discussion to the end. Yeah. So thank you guys.
Okay, thank you.
Thank
That's
you.
all.